So did you know that there are actually 5 major types of calcium channels in our bodies? We have the L, T, N, P, Q, and R types. Now, while they may sound similar, each one is unique with its own location and function. So in this video, let's get to know them all. Welcome once again, guys, to another exciting episode. This is your host, and I hope everyone is doing well. Before we begin, don't forget to hit the like button and subscribe to never miss out on future videos. All right, let's get started. Calcium plays a pivotal role in various physiological processes from muscle contraction to nerve impulse transmission. It does so by acting as a crucial secondary messenger. But how does this happen? Well, it all begins when a stimulus like a nerve impulse arrives at the cell membrane of a neuron or muscle cell. This triggers a cascade of events leading to the opening of specialized protein pores called voltage-gated calcium channels. When these channels open, it allows positively charged calcium ions to flow into the cell. This influx of calcium initiates a series of biochemical reactions that ultimately lead to the desired physiological response. For example, in muscle cells, this can be the start of a contraction, or in neurons, it could be the release of neurotransmitters. The primary role of calcium channels is to facilitate this controlled entry of calcium into cells, playing a vital role in maintaining homeostasis and ensuring proper cellular function. So if you want to know more about these amazing biological structures and the processes they control, keep watching. We're just getting started. First up, we have the L-type or long-lasting calcium channels. As the name suggests, L-type channels are known for their long-lasting openings. They are primarily found in 2 locations, cardiac muscle cells and smooth muscle cells. In cardiac muscle cells, they are abundantly located in the cell membranes of the sinoatrial node and the atrioventricular node, which are crucial for regulating the heart's rhythm. Within the cardiac muscle cells, they are found in the membranes of the sarcoplasmic reticulum, an organelle that plays a key role in storing and releasing calcium ions, which are essential for muscle contraction. The L-type channels are activated by an increase in membrane potential, meaning they open when the cell's internal electrical charge becomes more positive. This characteristic is shared by most voltage-gated ion channels, which are proteins embedded in the cell membrane that open or close in response to changes in the electrical potential across the membrane. Once activated, L-type channels allow calcium ions to flow into the cell, where they bind to specific proteins, triggering a series of events that lead to muscle contraction. This influx of calcium also plays a vital role in regulating the heart's pacemaking activity, directly influencing the heart rate. Interestingly, L-type calcium channels are the primary targets for a class of drugs known as calcium channel blockers, which are commonly used to treat high blood pressure, angina, and certain arrhythmias. These blockers selectively inhibit the activity of L-type channels, reducing the flow of calcium into cells and subsequently decreasing the force of muscle contractions. This effect is particularly beneficial in the heart, where it helps to lower blood pressure and improve blood flow. Next up, we have T-type or transient calcium channels. T-type calcium channels are named for their transient nature, which means they open and close quickly, allowing for a rapid influx of calcium into the cell. Unlike L-type channels, they are not found in cardiac muscle cells, but are predominantly located in neurons and certain endocrine cells. In neurons, T-type channels are found in the cell bodies and the dendrites, which are the structures involved in receiving signals from other neurons. Their primary role is to regulate low-threshold calcium currents, contributing to the pacemaker activity of neurons and the initiation of action potentials under specific conditions. Action potential is a wave of electrical activity that travels along the membrane of a neuron, allowing for the transmission of signals throughout the nervous system. T-type channels play a crucial role in the central pattern generator circuits in the spinal cord, contributing to rhythmic activities like walking and swimming. Additionally, they are involved in the regulation of neurotransmitter release at synapses, the junctions between neurons. By controlling the influx of calcium, T-type channels indirectly influence the communication between neurons. T-type channels are also important in the retina, where they are involved in the phototransduction process, the conversion of light energy into electrical signals. Here they contribute to the sensitivity of the retina to low levels of light. The activation of T-type channels in retinal neurons leads to the release of neurotransmitters that regulate the activity of downstream neurons, ultimately influencing our ability to see in dim light conditions. In contrast to the widespread use of calcium channel blockers for cardiovascular conditions, there are currently no clinically approved drugs that specifically target T-type channels. However, certain anticonvulsant and antidepressant medications are believed to modulate the activity of these channels, highlighting their potential as therapeutic targets for neurological disorders. Now let's talk about N-type or neuronal calcium channels. N-type calcium channels are the most abundant type of voltage-gated calcium channels found in neurons. As their name suggests, they are widely distributed throughout the nervous system with a high concentration in the brain, spinal cord, and peripheral nerves. Within neurons, N-type channels are primarily located at the nerve terminals, the ends of neurons where they form synapses with other neurons or muscle cells. These channels play a pivotal role in neurotransmitter release, the process by which neurons communicate with each other and with muscle cells. When an action potential reaches the nerve terminal, it causes the opening of N-type channels, allowing calcium to flow into the terminal. This influx of calcium triggers a cascade of events that ultimately leads to the fusion of synaptic vesicles filled with neurotransmitters to the presynaptic membrane. This fusion releases the neurotransmitters into the synaptic cleft, the small gap between the nerve terminal and the next cell, where they bind to specific receptors, initiating a response in the recipient cell. N-type channels are also involved in other important neuronal processes, including neuronal excitability, the ability of neurons to generate and propagate action potentials, and synaptic plasticity, the ability of synapses to strengthen or weaken over time. Synaptic plasticity is fundamental for learning and memory, and alterations in N-type channel function are thought to contribute to cognitive deficits in various neurological and psychiatric disorders. N-type channels are sensitive to a group of natural compounds called O-conotoxins, which are produced by the marine snail Conus geographus. These toxins have been explored as potential painkillers due to their ability to block N-type channels and inhibit neurotransmitter release in pain-transmitting neurons. Moving on, we have the PQ-type calcium channels. PQ-type calcium channels are also known as alpha-1A or alpha-1B subtypes, based on the molecular structure of their pore-forming alpha subunits. These channels are predominantly found in the central nervous system, particularly in the cerebellum, where they play a significant role in motor control and coordination. In the cerebellum, PQ-type channels are located in the Purkinje cells, a group of large neurons that are crucial for integrating and coordinating information from various sources to fine-tune motor movements. These channels contribute to the pacemaker activity of Purkinje cells, helping to regulate their rhythmic firing patterns, which are essential for motor coordination. PQ-type channels are also present in the dorsal root ganglion cells, which are sensory neurons responsible for transmitting pain and temperature sensations to the central nervous system. In these cells, they play a role in the release of substance P, a neuropeptide that modulates the perception of pain. Additionally, PQ-type channels are found in the hypothalamus, an area of the brain involved in hormone secretion and the regulation of body temperature. Here they contribute to the control of neuroendocrine functions. Notably, PQ-type channels are distinct from other neuronal calcium channels in their sensitivity to specific pharmacological agents. They are highly sensitive to the calcium channel blocker O-conotoxin GVIA, which has been explored as a potential therapeutic agent for neurological disorders characterized by abnormal neuronal activity. Finally, we have the R-type calcium channels. Our understanding of R-type calcium channels is still evolving, and they remain one of the least studied types of calcium channels. What we know so far is that they are widely expressed in the central nervous system, particularly in the brain, where they are found in regions associated with cognition and sensory processing. Like other types of calcium channels, R-type channels are activated by changes in the cell's membrane potential, allowing calcium ions to flow into the cell. However, the exact role of R-type channels in neuronal function is still not fully understood. Some studies suggest that they might play a role in neurotransmitter release, similar to N-type channels, but at different synapses or in response to different stimuli. Others suggest that R-type channels may be involved in regulating gene expression, contributing to long-term changes in neuronal activity and function. R-type channels are also thought to be involved in synaptic plasticity, which is the ability of synapses to strengthen or weaken over time. This process is fundamental for learning and memory, and dysregulation of R-type channels may contribute to cognitive deficits in neurological disorders. While research on R-type channels is still ongoing, they have emerged as potential therapeutic targets for neurological disorders characterized by abnormal neuronal activity. Several classes of drugs that modulate the activity of R-type channels have shown promise in preclinical studies, suggesting that they could be useful for treating conditions such as epilepsy, neuropathic pain, and schizophrenia. So there you have it, a comprehensive overview of the 5 major types of calcium channels in the human body. Each type is unique, serving distinct roles in various physiological processes from muscle contraction to neurotransmitter release. The study of these channels not only deepens our understanding of normal cellular function, but also provides insights into the mechanisms underlying various diseases. Moreover, exploring the intricacies of these calcium channels holds immense potential for the development of novel therapeutic strategies for a wide range of conditions. As research progresses, we can anticipate a better understanding of these intricate biological processes and the development of targeted therapies for conditions that affect them. If you find this video helpful, please leave a comment down below and share it with your friends. And if you haven't already, please take a moment to subscribe and hit the notification bell icon. Thank you for watching, and until next time, stay healthy and happy.